Weinberger.
Thank you very much. Dear colleagues, I would like to start by thanking the organizers for the invitation. Contractile patches, wishes and visions. Okay, I'm neither a pediatric cardiologist nor a cardiac surgeon. Wishes, specifically designed for pediatric patients, are difficult for me to anticipate. And I would like to use the opportunity to give this talk to present what is doable, what is technically doable, what we aim for in the adult heart, but I would also like to use it as an opportunity to start the conversation about wishes. When we talk about contractile patches, we talk about contractile cells, cardiomyocytes, and this represents a problem because early after birth, the heart loses the ability to regenerate. There are hardly any new cardiomyocytes formed in the postnatal life. We currently estimate that about 1% of cardiomyocytes are newly formed per year. And this number might increase slightly in a myocardial injury, as a myocardial infarction, or as seen here in an experimental model of LAD ligation. However, even in these situations, no significant number of new myocytes is formed, which means we can't just take a little piece of heart tissue, isolate the myocytes, make them proliferate, and then use them to generate a contractile patch. The situation is slightly different in the immediate postnatal period. When you injure a neonatal heart, it will regrow with only little scarring. This was initially described in mice. There is anecdotal data in humans, and just 2 or 3 weeks ago, 2 large studies were published that showed that this also exists in pigs, indicating that it might exist in humans as well. However, this window is short, and within 1 or 2 days after birth, it closes. Therefore, we need another cell type. We need a cell type that enables us to generate a large number of cardiomyocytes, basically an indefinite number of human cardiomyocytes. And the only cell type that is able to allow us the differentiation of human cardiomyocytes are pluripotent stem cells, either embryonic stem cells or induced pluripotent stem cells. And then, by recapitulating the embryonic development in a dish, we generate cells of mesodermal fate, later on cardiac progenitors, and eventually cardiomyocytes. We use induced pluripotent stem cells in the lab. Those were initially described by Shinja Yamanaka, who then later on got the Nobel Prize for this discovery. Induced pluripotent stem cells are reprogrammed from dermal fibroblasts initially, from somatic cells. We nowadays can also reprogram them from leukocytes. So by overexpressing transcription factors, we drive those somatic cells to a pluripotent fate and then differentiate them to cardiomyocytes. The real-life scenario looks like this. That's a pluripotent stem cell colony. That's an induced pluripotent stem cell colony. And then we recapitulate embryonic development. And with the help of cytokines, create these embryoid bodies consisting of mesodermal cells, cardiac progenitors, and eventually cardiomyocytes. And it's rather easy to see that those are cardiomyocytes because they beat. They're immature and they show spontaneous beating in a dish. So we're able to differentiate a large number of human cardiomyocytes, and we're able to use those to generate contractile patches. There are different ways to do it. We use a hydrogen-based approach, meaning we entrap those cells in a fibrin matrix, and we call these contractile patches engineered heart tissue, or short EHTs. And then after about a week or 10 days in culture, they coherently start to beat. There are mainly 3 purposes that we use those EHTs for. First is disease modeling, second one is drug screening, and then there is the third one, which is regenerative medicine. So my first take-home message is, yes, we're able to generate human contractile patches. For regenerative purposes, we have started with these stripe format EHTs. However, we're able to generate them in different shapes. So we're able to generate these tube-shaped structures, look like this. This is something that we've just started and haven't really followed up. And as we mainly focus on the adult heart, we create these mesh-structured patches that look like this. And again, can you start the video, please? Thank you. After about a week in culture, they coherently start to beat. So, second point, we're able to engineer those patches in different shapes. As I said, we're mainly interested in the adult heart, and we currently use those mesh-structured engineered heart tissues in various transplantation models. And this is a histology of one of those patches, stained for phalloidine, stained with phalloidine, which stains the actin cytoskeleton of the cells. And I would like to draw your attention to two characteristics. First one is cell density. These patches consist of densely packed myocytes. So we see a rather homogeneous cell distribution within those patches. That's the first point. The second point is they mature. Stem cell-derived myocytes, initially after differentiation, are immature. This is basically true for all stem cell-derived cells. Within the three-dimensional construct, they mature, and I think this is already visible in this low magnification. They align along these force lines, and it's probably even better to see in a higher magnification those cardiomyocytes that initially looked roundish, elongate, and become something that looks like, that resembles an adult cardiomyocyte with this elongated rod-shaped form. And this is an example of a transplantation in a small animal model. This is a guinea pig heart. So what we do is we initially injure the guinea pig heart, and this is seen here. So this is dystrophin staining, meaning that muscle is stained in brown, and then there is this large injury of the left ventricular wall. And about a week after injury, we epicardially transplant one of those mesh-structured patches. And then 4 weeks later, we perform histology, and this is shown here. And that's the newly generated human myocardium. And as a proof that it indeed is human myocardium, we've done staining for a human marker, in this case human Q80 in an adjacent section. So these patches survive transplantation, but they do not only survive transplantation, they mature even further. So this is a histology of one of the grafts. And you can see that this graft consists of densely packed myocytes. And in a high magnification, you can see they still do not look like native myocardium. However, they resemble something that looks like adult myocardium. The cells are still smaller. They are larger than in the three-dimensional construct, so they grow. But they're still smaller than the adult cells, and they are not as properly aligned. However, they do mature in the construct and even further after transplantation. Why do I strengthen this point? Why do I strengthen this point about maturation? Well, one of the limitations so far clearly is force development. Even the best stem cell-derived constructs develop lesser force than the native myocardium, by far. So even the best constructs that we're able to engineer do not reach the force that native myocardium is able to reach. So this clearly is so far a limitation. A second limitation is upscaling. It is easy, technically easy, to upscale these constructs in 2 dimensions, which is length, width, but then there is a third dimension, which is height, or let's call it thickness. And as I said, these constructs, these cells, during culture, they do rely on diffusion. So if we just increase thickness of these constructs, which is easily doable, we just use more matrix, this will end in a cell-free necrotic core, which probably doesn't help. So thickness is a limitation. However, biology might help us to overcome it. This again is a hard 4 weeks after transplantation. In this case, we have transplanted 2 of these stripe-formed engineered heart tissues. At the time of transplantation, they have a diameter of about 800 microns. So if I project those onto the graft, you can see that just within 4 weeks, so 4 weeks after transplantation, they have increased in size. And there are 2 possibilities, 2 explanations for this. One is hypertrophy, so the individual cell becoming larger, bigger. And this happens, but it definitely does not account for this increase. And then there's a second explanation, which is hyperplasia. And we think this is something that also happened. So we think that after transplantation, the stem cell-derived myocytes re-enter the cell cycle and proliferate. This is one indication. We have more indications for this. This animal has received BRDU after transplantation over a 1-week period. BRDU is a nucleotide analog. So whenever a cell divides, proliferates, it has to synthesize DNA, and therefore it needs nucleotide analogs. So proliferating cells incorporate BRDU in the nucleus, and we're then able to trace it. So every nucleus here is stained in red, and every nucleus that has performed DNA synthesis as an indication of proliferation is also blue. And then in green we stain for a cardiac marker, and this is alpha-actinin in this case. So this is the graft. These are nuclei that have not performed DNA synthesis, but we clearly see nuclei that have performed DNA synthesis. Again, another indication for ongoing cell proliferation. So we think that they indeed proliferate, and this proliferation might help to overcome the limitation in height, thickness. So where are we? Are we ready for a clinical trial? Well, stem cell or stem cell-derived cells are ready for clinical trials. There are some clinical trials already that have been performed with embryonic derived cells. The first ones were for macular degeneration, and this is the first study that was performed in Japan using iPSC, induced pluripotent stem cell derived cells. And the first cardiac study with iPS cells will start probably this year or early next year, also in Japan. We in the lab have a stem cell line, an induced pluripotent stem cell line that has been reprogrammed under good manufacturing practices by the NIH and New York Stem Cell Foundation, and we were able to generate human cardiac patches in clinical grade conditions. So indeed, I think we're in a time where stem cell derived approaches translate from basic research towards first clinical trials, which is exciting. Okay, coming back to the beginning, wishes and visions. This is from a review already almost 10 years ago, where they envisioned that we're able to generate a new ventricle for patients with hypoplastic left heart syndrome, or to engineer a septum for a patient with an univentricular heart. That's a bold vision. And I don't see this in the immediate future. However, we have some ideas how to get there. And as I said in the beginning, I really would like to use this presentation as an offer to start a conversation. And with this, I would like to thank you for your attention. This is obviously a team effort, and this is just the core of the team in Hamburg. We're not the only ones working on these cardiac patches. There are other groups in Germany and there are other groups worldwide. But as I said, I think exciting times, and I think that we hopefully will see this in first clinical trials within the next year. Thank you very much.
Thank you very much for this innovative and excellent talk. But first of all, I have to welcome Professor Lange, the name giver of this symposium. Welcome, sir.